Okay, today we're going to talk about apoptosis. Last couple we talked about cell cycle, and now we start getting into cell death. And if you are interested in doing some of your basic biology reading, this is essential biology. Use this keyword for PubMed. If you are looking for a chapter in a book, go to PubMed books and then type in apoptosis. You will have chapter 18 for your reference. Okay? And if you like Lodish's textbook, which is molecular cell biology, you should look for chapter 19 and 21. 19 is about cell cycle. Chapter 21 is about cancer biology. It's a stem cell. And this one is molecular biology of cell. And it's on chapter 18, which is, this textbook is a more advanced than essential cell biology. This one is like a short version of this one. Okay? So we are going to talk about cell lifespan and cell death. And what's the differences between apoptosis and necrosis? Do you know what is necrosis?
Yeah.
Oh, okay. And the significance of apoptosis in development and in morphological change. Finally, we're going to talk about the signaling of apoptosis, which is the mechanism. It has two pathways. One is intrinsic, one is extrinsic. Well, let me ask a question. Do you have apoptosis this lecture before when you were undergrad?
No, I don't.
No? No? They didn't talk about apoptosis or cell death in your biology class? Just shoot it, right? Okay. Life and death, just like yin and yang. Okay? And look at these two terminology, apoptosis and mitosis. So number one, the root of the word is SIS, meaning the phenomenon. Okay? And mitosis is something you observe. When you look under the microscope, there are thread-like features happening in this cell, and then this cell will become two cells. Okay. So that's something that observed under the microscope. The same thing in the apoptosis. In this H&E slide, or this cell, the one is smaller and darker. This is H&E standing. The darker blue stands for nuclei. The normal cell should have a morphology looking like this. Okay? Smaller one, darker one, meaning your nucleus is condensed. And that's one of the morphological change when the cell undergo apoptosis. Okay? So both life and death, both terminology coming from when scientists observing the microscopic phenotype and then give this word. Okay, so the lifespan is different for cell in our body. For example, in human body, the neutrophil. You know what's neutrophil? That's neutrophil. Okay, that's neutrophil in circulation. That's neutrophil in tissue. So neutrophil in tissue, the lifespan is different from neutrophil in circulation. Neutrophil in circulation only have 6 to 7 hours. And neutrophil in tissue can live for 4 days. Do you know what's the difference?
Differences in what?
Why neutrophil? It's all coming from the same source. And they all call neutrophil. But if they go to the tissue, they live longer. If they stay in the circulation, the lifespan is shorter.
Is it because it's more mobile in the circulation system? Because they live within
more than normal condition?
Well, in normal condition, the neutrophil is in the blood circulation, right? When does it go to the tissue?
When there's tissue damage?
Yes.
Ah.
When the immune system needs its help, okay, it goes to the tissue. What happened in the tissue? Some cell got damaged and they will secrete cytokine and call for help. The signal coming from the tissue going into the blood circulation, and the neutrophil will be attracted to the tissue because the environment is different. It has cytokine, it has other stimulation. Therefore, the neutrophil in the tissue live longer. Think about, you have a lot of food around you, so the neutrophil has a lot of food in here. Okay. And for the red blood cell, normally it's about 120 days. So when we try to culture white blood cell, leukocyte, okay, from one of the blood sample from patient, the number one difficulty is to remove the RBC from the sample, because normally there is a trace of red blood cell contamination in your white blood cell fraction, and it's really hard to get rid of the RBC, and the only way is wait until they die, giving stimulation to the white blood cell but not the growth factor for the red blood cell. The brain neuron has a lifespan of 50 to 100 years. Okay, so in the same organism, human, you have cells in different tissue that have different lifespan. Why? So for any cell has really long lifespan, for example, brain or neuron, even your cardiomyocyte. If the lifespan is longer, meaning I don't have to replace it for every 100 years, right? So next to that tissue, you don't have a lot of stem cells to repair it. Okay? So treatment of any neuronal condition is difficult because we don't have a lot of stem cells for all these neural tissue. Exactly. So we have to do it in vitro to convert or differentiate other stem cell into neuronal cell for transplant purpose. And the same thing for your cardiomyocyte, because the cardiomyocyte can live longer. Your red blood cell, your neutrophil, lifespan is really short. It's like 120 days, really long, like 4 days, 6 or 7 hours. That's why you have bone marrow replacing as a source of your stem cell for your blood. Okay? And it's get regenerate every 4 days, every 2 months, 4 months. Okay, got it. So the resource of your stem cell is proportional to, or is reverse proportional to the lifespan of the cell. And that's special staining for neuronal cell. That's for my cell biology class. So the lifespan process includes birth, death, and programmed cell death. Okay, so all these three have different mechanisms. The picture on your right-hand side is the process of apoptosis. Apoptosis, not programmed cell death. Okay. So the definition for birth is if the cells undergo mitosis or meiosis. Definition for death is the process of apoptosis or necrosis. We'll tell the differences. And actually, we'll talk about a lot of different death today. Okay. Programmed cell death is a biological phenomenon, and it was coined by William Curie in 1972. Apoptosis, this terminology and phenomena was determined in 1990 or late 1980s. That was way after programmed cell death. There is a relationship between apoptosis and programmed cell death, but there is no, they are not the same thing. And I will try to describe the differences between these two using a diagram later. Not now. Later. Okay. Oh, now here's the stuff. So apoptosis is a morphological description of dying cell. It's dying. Okay. Programmed cell death, or PCD, is a term used to describe the cell that die at predictable time and place during the development. So it was originally coming from developmental biology, when scientists find that there are cells died in the process of development. Okay. That's the whole process is programmed cell death, and the final phenomenon, the final phenotype is apoptosis. So here, if this is our cell cycle of G0, G1 transition, so when the cell is not doing the cell cycle, exit the cell cycle, stay in the G0 state, right? It's quiescent, meaning it's doing nothing, sitting there. Or it's just doing nothing. One day, if the environment is too poor, or there is no supporting growth factor to support to stay in quiescent, well, you will go to the death pathway. Okay? From quiescent to death, the process is called senescence, aging. The whole process is called aging, because once you activate the senescence process, it's no way to go back. When you get old, you don't get young, okay? There's no such thing to go reverse way, okay? And right before the death, you started the apoptosis mechanism. Remember, that's the morphological change of dying cell. So something happened to make the dying cell has characteristic changes on its phenotype, and that we say is a apoptotic cell. And the end of apoptosis is the death. Okay? So somewhere during the senescence process, the organism can say, well, this cell should go old and then go dying. Okay? So the whole predictable time and places is determined by the programmed cell death. It's a more complicated mechanism. Got it?
Do you mean by the aging, it's like not suddenly die, or?
Well, aging is the whole process from process, the whole process. It's senescence, the whole process. So there are different experimental evaluation or assay for senescence, and different experiment for apoptosis, different experiment for programmed cell death. It's different. Okay? So you can imagine there are many different mechanisms activated for programmed cell death. At the same time, those mechanisms will affecting apoptosis or senescence process. Okay, so programmed cell death is more complicated. Any question? You want to share? Question? You can ask. No? No? Okay. Well, because a lot of people get confused by quiescent and senescence. They'll put equal in here. You'll put apoptosis and programmed cell death equal. But there are differences between all these terminologies, okay? Now story time. These three scientists, they got their Nobel Prize because they are studying cell death in 2002. So the one on the left-hand side is the first person who coined the word apoptosis, Cindy Brenner. The one on the right-hand side is the scientist from South Africa. And Dr. Sultan is from England. And then Robert Horvitz is from United States. And Cindy Brenner is the scientist. I actually went to his lecture once.
Oh, really?
In 1992, because I had no idea what is apoptosis. It was something really new. And he was telling us about how he did the experiment with C. elegans. Do you know what is C. elegans?
Ligand.
C. elegans, not like a
C. elegans?
C. elegans is a worm, like kind of transparent, and the C. elegans. Oh, I should have C. elegans here. Dr. Brenner, Cindy Brenner, he do you know what is that? What is that in Indonesian?
We don't know. It's like very small.
线虫 in Chinese. 线虫. It's an animal model for non-molecular biology study. Yeah.
Yeah, is that a ringworm?
It's a worm. It's not a ringworm. It's not.
She's a worm.
It's a worm. And
Can we see it in our eyes? No.
You can see it in a petri dish because it eats E. coli as a food.
Oh.
And so about 20 years ago, you should ask Gary. He was working with C. elegans this summer.
Really?
Yeah, he did. Yeah, he had a lot of fun. And C. elegans was an animal model because one of his favorite food is E. coli. So 20 years ago, there are a whole bunch of RNAi study or any gene expression study using C. elegans, because all you need to do is to use the plasmid that express the protein or can generate RNAi fragment and feed it to a C. elegans. You can study the transcriptome of the whole C. elegans. That was before the RNA-seq was popular. You can use C. elegans to study all those RNAi stuff. So this scientist, Cindy Brenner, the first scientist using C. elegans to study, but he was in chemistry major, and actually he was studying in nerve cell, and he was looking at when C. elegans in the process of development, he was counting how many cells this organism should have. So the adult C. elegans should have 1,090 cells. But during the developmental process, there are 131 cells died in the process. If he tried to generate mutation, try to stop dying of these 131 cells, it leads to abnormal growth of this nerve innervation. Okay, so the first conclusion he has from C. elegans experiment is like this nerve cell was the organism make decision to have all these cells die during this developmental time after the neuroimmunization process is done. Okay, so you need a lot of neuroimmunization during the development, allow the nerve stem cell to grow. Once they grow to certain size or when they mature, they will compete the growth factor. Competition of trying to fight for the growth factor will cause the death of those interconnecting nerve cells survivor. So those cells will die. Without dying, all the other nerve cells will be immature because they don't have enough nutrient, they don't have enough growth factor. And that's the conclusion he had for studying the C. elegans. He didn't mean it to, study apoptosis. He just found some cell, they must die at that time point. Without dying, your nerve cell will not mature. Okay? So that's his contribution. And Dr. Horvitz, he's the one generate all these mutation called CED, C. elegans death gene, CED. So we learned CEC last week, and now this mutant is called CED. And he's the Dr. Horvitz, this guy here. He actually is the one trying to study all the mechanisms of apoptosis, okay, and all the molecular biology assay. And Dr. Sultan, I forgot what he did. He's important, but I just forgot what he did. Do I have Sultan's? Oh, Sultan is interesting. Oh, his contribution. He sequenced the genome of the C. elegans.
Oh.
Yeah. Okay. Very important, but nothing to do with apoptosis. It's helping Dr. Horvitz to study the molecular biology. Okay? And Sidney Brenner, well, he passed away in 2019, but I think it's in 2000. He moved to Singapore because Singapore was trying to establish the #1 molecular biology research center in the world. It's called Center. So they have a Center in Singapore. And Sidney Brenner was there as the first director of the center. Sidney Brenner. Oh, he's also Jewish. We have a lot of Jewish scientists. Yeah. And he's also a doctor. He graduated from Oxford, and I think he graduated from Cambridge. Okay. So let's look at two major types of cell death. One is apoptosis, one is necrosis. So apoptosis is programmed cell death, meaning you know when to die, you know where to die.
Wow.
That means suicide, right?
Okay. Suicide.
Yeah, suicide.
In a good way.
Yeah.
For a good way.
In a good way.
For good.
And
Yes, for good.
necrosis is accidental cell death. Well, the cell wasn't going to die, but somebody murdered it. Okay? Somebody killed it. That's accidental. Yeah. So if the cell died with accidentally die, the major phenotype of this cell is just like you drop a piece of glass. It's going to break into fragments right away. Everything inside the cell will be spelling out. Okay? For apoptosis, if the cell died like necrosis, this the content from the neighboring cell, which is bad for the organism. Therefore, the mechanism of apoptosis try to keep it as small, the dying cell as small as possible. Allow the macrophage to engulf it. Okay, so they won't disturb the neighboring cell. Okay, just one cell die is fine, but necrosis will affect the neighboring cell. In normal or in a perfect situation, necrosis and apoptosis should not be there at the same time. But that's an ideal situation. But our tissue has a lot of stimulation and a lot of accident going on. So sometimes in an aging cell, you will see some necrosis spot going on. Okay? Okay, so that's well. This is a if we talk about morphological change, apoptosis. Oh, these two are just lipo, the tungsten filament lipo. Apoptosis is like the filament is getting thinner and broken. When the filament is getting thinner, the energy for the lipo is getting smaller. So the dying is like it's gradually happen, and at the end of the death, the whole lipo is not it's not working anymore. For necrosis, the lipo is like being hit by, the outer force. Okay, so everything breaks, but the tungsten filament is still on. Still on. Yeah, it's going to go away. The lighting is going to go away when the oxidation of the filament is complete. Do you know what is inside the lipo? It's inner gas. Yeah, that's why it's not going to get oxidized too fast. I'm not physics. No physics here. Okay, so necrosis usually is killing by injury or outer force, and apoptosis is something committed by the cell. Okay, one is a passive process, one is an active process. Passive process meaning this person or this cell does nothing, just standing there. Somebody killed it. Okay, so the energy is coming from somebody else. It's not from this cell. For apoptosis in active process, the apoptosis requires ATP. So it's going to use some energy from the cell to achieve the result. Okay? This energy is coming from somebody else. Okay? So that's the major differences. And the necrosis will lead to inflammation. That's why it's causing damage of the neighboring cell and the environment. Apoptosis has no inflammation. So that's one major feature. When we're looking at the tissue, whether it's apoptotic tissue or inflammatory tissue, we're looking for inflammation. So what happens in the tissue when you have inflammation? You see a lot of leukocytes coming into the injured site. So there are a lot of blue dots inside the leukocyte or T cell or B cell inside the tissue. For apoptotic cell, we don't see all these leukocyte infiltration into the T cell, into the tissue. Okay? Do you know what is infiltration? Infiltrate. Tissue will infiltrate by the immune cell. Yes. So something happened in the tissue, they send out the cytokine, the immune cells coming from the blood vessel, they cross the blood vessel and come to the tissue. It's called infiltration, which is not a normal situation. Okay? That's a terminology for medical technologists. Okay? So every day, approximately 5% of an organism undergo apoptosis. That's a normal cell number, or normal fraction of apoptotic cell. And timing is about 15 to 120 minutes. Meaning after this class, some of your cell is dead already, right? That's normal. And it's not doing any harm to you, right? Well, yeah, you're getting older. Okay, there are several types of cell death. Apoptosis belongs to programmed cell death. Okay, and the one on the left-hand corner is programmed cell death, but they are not apoptotic. So that's programmed cell death, and this one is non-programmed cell death. About 50% of the cell deaths belong to necrosis. Okay, that's proportion. And apoptosis is about 1/4, maybe less than 1/4, maybe 1/5. The other type of cell death, which is important, is autophagy. We'll talk about autophagy and mitosis. The other one is the mitochondria part. You have mitosis, but the correct name, starting a couple years ago, is called mitophagy. Mitophagy instead of mitosis. Yeah, that's the late trick. Mitosis.
Mitopsis.
It means mitochondria undergo apoptosis, but recently, this couple years, they found the process is not apoptosis. It's more like autophagy. Yeah, it's like a phagocytosis. That's interesting. If you are interested in mitochondria's mitophagy, you can ask Al. She did a presentation on that. Yeah. Alvin, in our lab?
Yeah. I saw her.
You saw her?
In front of 7-Eleven.
Yeah. Okay. So let's the type of cell that we'll talk about necrosis and apoptosis. So I will just let you know what is autophagy, what is mitosis, and what is pyroptosis. Let's look at autophagy. Autophagy is degradation of cellular component within dying cell. So the cell is dying, but autophagy is the mechanism inside the dying cell. So not whole cell, not the whole cell. Okay, apoptosis is morphological changes of the whole cell. Okay, that's the differences. It begins with a cytoplasmic material within the phagosome. So it's a process involving phagocytosis of inside the cell, okay? Not the whole cell. And under control of GTP, phosphatidylinositol kinase. So any kinase is to do phosphorylation of the target protein, and that causes structural changes, allow the membrane to engulf the damaged part. Okay? So let's look at autophagy. Autophagy. 自噬. Do you know what this word is? The second one? Eating. Eating? Eating. 是. The first word. Yes. Self. Eat yourself. Autophagy meaning eating yourself. Okay? 自噬. Eating of self. 自噬. And that's the translation, autophagy translation in Chinese, 自噬. Do we have this topic in biology class? Not the cell biology. I know I have to talk about it, but do we have this in biology class? They don't talk about this mechanism. So it's about how lysosomes try to degrade the use material or damage material inside the cell. And it initiate with the membrane. The membrane could be from the vesicle, the membrane could be from your cytoplasmic membrane, inward of the cytoplasmic membrane. And there are 3 different types of autophagy, okay? We'll talk about this later. Well, start with membrane, and then membrane will recruit factor that involve in phagocytosis, okay? And calling them to come to the membrane and help to become a autophagosome. This autophagosome will fuse with lysosome. The lysosome will destroy whatever inside the autophagosome. Okay? So actually, the most important part is here. Imagine this is like endocytosis, right? But when material is coming from outside the cell, endocytosis inside the cell, when it fuses with the lysosome, lysosome will take care of whatever is coming in from outside. Okay, so one is macro autophagy, and this is a chaperone-mediated autophagy. So chaperone, our Hisham protein. Anything that is misfolded can be sent for autophagy instead of using proteasome. Remember, if you have unfolded protein inside the ER, the chaperone inside the ER will do ubiquitination and send it to the proteasome. But what happened inside the cytoplasm Hisham protein will send it to the lysosome for autophagy process.
Is it the protein of HSF also involved in this?
HSC,
It is.
Hisham protein 70. Okay.
I've also heard about that heat shock factor 1.
Hisham factor 1, that's different things.
Okay.
Chaperone, the major core component of the chaperone system is HSP70. If the cytoplasmic form is stated in HSC, so HSC70 has SSA1, SSA2. It's complicated. Don't ask me this question. But they do have other partner for the whole system. It's called in prokaryotic system, HSP70 is DNA K, and the other partners are DNA J and group E. When we come to the eukaryotic system, DNA K is HSP70. They also have DNA J, which is SSP40. And the group E is HSP1E, I forgot what's the molecular for that. So they're the whole system. And the partner of the other Hisham protein is to accelerate the process. Without HSP70, you don't have this chaperone's activity, meaning attenuate the folding or helping autophagy process. Okay? So this is too complicated. I'm not going to go into detail. It's not necessary, but just let you know all these facts, they are 17 genes for initiation of the autophagosome. Okay? The engulfing of the membrane, and then to isolate all those bad material inside the autophagosome. And that requires 17 or maybe more than that factor. Gene that is has short like ATG, meaning autophagy pathways gene. Because this ATG. It's not that important. Okay. The second one is apoptosis. That's the invasion of living cell into another cell cytoplasm, meaning one cell eating another cell alive. Okay, and that happened in or cell-in-cell structure. That happened in a cancer cell. Yeah, the cancer cell eat, and it's called apoptosis. The Chinese word, the first two words are 细胞 cell. 细胞.
Wow.
Last two words, 互视 eat each other. 视 eating, right? 互 meaning 互. What is English in 互? 互 each other. 互相才是 each other, right? 互相. So they take 互相. Have you heard 互相帮忙? You help each other. 互相帮忙 help, right? Help each other. 互相帮忙. The first word 互视 互 each other.
Why are you so doubtful?
I'm not very good in Chinese. So remember, this is not apoptosis in the absence of apoptosis. Many types of developmental cell deaths can still occur by other mechanisms. In especially mammalian epithelial cell, we'll talk about how the epithelial cell inside the lumen, they get shed off. I mean, they die. And they have different type of dying. One of them is apoptosis. We'll talk about that later. When they get detached, the epithelial cell get detached from their basal membrane and then fall into the lumen. It's called anoicus. It's a different terminology. Just as you know, they have different terminologies. Yeah. That's anoicus. Okay, detachment of this epithelial cell inside the lumen. Think about this: the lumen of your intestine. Okay. The reason the cell get detached because there is no attachment to the neighboring cell or basement membrane by the adhesion molecule. So one of the problems could be the loss of the adhesion molecule. The second problem is maybe there is a turnover rate for the adhesion molecule. Okay. Or number three, there's damage. That's apoptosis, meaning you see cell eating another cell. So inside the luminous cell, if you have rapid proliferation like this, it's behaving like cancer cell. But the purpose of proliferation inside the luminal epithelial cell is to replace the old cell or damaged cell. So sometimes in the proliferative cell, you will see apoptosis, a cell-eating cell things. And sometimes you will have necrosis or autophagy. These are 4 different types of cells that commonly see in our lumen of intestine or GI tract, or maybe your respiratory tract too. Yeah. So apoptosis is also detachment from the ECM and by all these mechanisms. And the detachment will initiate cell death process. One of them is apoptosis. So all those apoptosis or anoicus or apoptosis is referring to the death of luminal cell, and it's either they could survive or they could die. Dying and surviving require different pathway. the one can get escape is by cell-eating cell. Yeah, they can survive. But if it's get autophagy or get fusion by the lysosome, the cell will die. So it depends, really depends. This is the experimental apoptosis using breast cancer cell. And MCF7 is a breast cancer cell one. And that's the process of apoptosis on the right-hand side. Because they are doing rapid proliferation. And in order for them to survive, they're going to eat each other. Let me check. This one here. Looks like a monster, right? I don't like it at all. And the reason the cell is going to eat the cells because we're using taxol to kill them. In order to survive, they activate the apoptosis mechanism. Okay. So last one is apoptosis. Apoptosis is followed by the inflammation. It's because the cell die because of bacterial infection. Mainly. So you will act since it's a result from bacterial infection, you will have inflammation. But at the same time, it activate caspase 1. Caspase 1 is one of the members in apoptosis. So this is a multiple mechanism happening inside in a apoptosis. It has apoptosis and apoptosis happening at the same time. And we will talk about how you make the differentiation between them. Okay? For apoptosis, it has everything you see in apoptosis except DNF laddering fragmentation phenomenon. They don't have this. They don't. Okay? Apoptosis do not have any information. That's the two major differences. Okay? We'll talk about all those experimental assay for apoptosis. Okay? So, let me see. 2点. Apoptosis, the definition for apoptosis is the capacity of a cell respond to stimuli by initiating pathway lead to its death by a characteristic set of reaction. It's a characteristic set of reaction. And the apple means off. Apoptosis means falling, just like the leaves falling from the tree. So in apoptosis, what does a stem cell look like? They look different, right? So the major characteristic for apoptosis, DNA fragmentation, chromatin condensation, and fragment of nucleus and cell membrane, and all the other part of cell membrane. For the necrosis, all the content inside the cell will burst out. So you will see cells swell and then burst out. Okay, so this is the four major features of the apoptotic cell. A is the shrinking of cytoplasm. So you will see the cell is no longer round and smooth, okay, due to the lamin and actin filament cleaved by your caspase enzyme, and that no longer hold the cell structure in the normal situation. So you will see shrinking of the cytoplasm and kind of unsmooth cell membrane. B is the nuclear condensation right here. Because the breaking down of a chromatin and the nuclear structure. So the DNA will be aggregated at one spot. Everything just tangling at the same spot. Okay, and that causes the condensation of the nuclei. The third feature is the packaging of cell into a form easily engulfed by the macrophage. So the dead cell will be cleared by the macrophage. In order for the macrophage to engulf the cell, he has to pack himself. It's as small as possible. When he's doing packaging, something is happening on the cell membrane. Okay, so the cell membrane will be disorganized, and some of the factor in the cytoplasm will be flipped out because you are trying to shrink the cell, and the cell membrane is disorganized. So once it was inside, the factor was inside the cell becoming outside. Once sometimes in normal situation, the factor that is outside becoming inside. So it's disorganized. At the end, you will see blabbing. This is called blabbing situation. Blabbing meaning you have blister on your wound, and a blister looks like this. It's like a blister. It's something coming out, a small apoptotic body coming out from the cell, getting smaller and smaller, easier for the macrophage to get engulfed. Okay, so that's the 4 feature for apoptosis. So that's blabbing. The one on the left-hand side is the TEM. Do you know what is TEM? The one on the right-hand side is the SEM. So SEM give you all those information on the cell surface, and TEM has required much stronger energy to penetrate the cell. So you'll see all the detail inside the cell. This one you don't see the detail, but you do see the surface. So here is no. It depends on your experiment. Sometimes the SEM give you more detail. Sometimes you want to see the content of the cell, you need TEM. Yeah. It's just a different electronic microscope. So when the cell die, the apoptotic cell express eat me signal outside of the cell membrane, and that signal will tell macrophage to eat it. Okay? Otherwise, the macrophage doesn't know which one they should eat. What happens is, in normal cell, the phosphatidylserine is restricted to the inside of the cytoplasm. Okay, so that's the outer cell. That's it. That's cytoplasm fraction. And when the cell go becoming apoptotic,
They go inside.
yes, they go inside out. So the signal of the phosphatidylserine will be coming outside, and that's a signal for macrophage. So the macrophage will recognize the signal, binds to it, and then engulf it. So that's one of, for the apoptosis, you are looking at cell shrinking, nuclear condensation, appearing of apoptotic body, and the blabbing, and annexin 5 and PI. I'll talk about this, and then no inflammation. Okay. For necrosis, cell swell, nuclear disintegration, and then membrane is dissolution. The annexin 5 and PI are all positive. So what is annexin 5? What is PI? What is PI? PI is a stain. PI is a stain, a chemical. but that chemical is equivalent to lithium bromide, which will bind to the nucleic acid. Okay, so if you look at, if you see anything like say PI, meaning it's binding to DNA or RNA. Where is the DNA and RNA? Inside the nucleus. So in order to allows the compound binds to DNA, it must go through the cell membrane and the nuclear membrane, then go into the nucleus, right? It binds to DNA. So that's one way to test whether your cell membrane is integrated or not. Okay? So on the left-hand side is the normal cell. On the right-hand side is early apoptotic cell. So this one is apoptosis. You have your phosphatidylserine that is outside. And phosphatidylserine, the molecule that's in pink, okay, that will bind to NSM5. NSM5 is just a molecule that will bind to phosphatidylserine. And if the cell is undergo apoptosis, you will see a green fluorescent, right? But do you see PI? At early apoptotic cell, the cell membrane still kind of intact. You don't see PI. At the end of the apoptosis, when everything gets condensed and packed into a small apoptotic body, then you can then the PI can go into the cell membrane and go into the nucleus. Okay, so PI is used to differentiate the timing of apoptosis. PI is also can differentiate whether the cell is undergo apoptosis or necrosis. What happens in the necrosis? Everything bursts out. So the membrane is not one thing. Okay? So PI can go in there at early stage of cell death. Okay. So we can use flow cytometry. Do you know what is flow cytometry?
The machine that really tests the information about how cells break.
How cell what?
Gene expression is also able to be.
No gene expression. No gene expression. Cell cytometry is the machine that detects the different sizes of a cell or different complexity of cell. Complexity means any grain structure inside the cell using the laser. The flow cytometry can also detect fluorescent signal that is outside of the cell, not inside, outside of the cell. So if you wanted to differentiate T cell from the B cell, okay, it's all leukocyte, they probably all look alike, and they probably about the same size, but the cell membrane has different marker or different protein for T cell and B cell. You can use antibody with fluorescent tag to differentiate T cell and B cell and passing it through flow cytometry to allow you to know how many T cells in here, how many B cells in here. You can also force them to separate. You can isolate the pure T cell or B cell using flow cytometry. Okay.
They're also able to detect the amount of cells.
Yes.
Right.
It counts about 10,000 cells per minute. It counts. And it can maybe more than that because I usually do 30,000 cells per minute. Yeah, it's really fast. But the assay is it's like antibody and antigen binding because you need to add antibody to the live cell and allow the antibody binds to the cell surface, and you can do your flow cytometry. So that's the result from the flow cytometry. I will teach you how to read the diagram, but not today, okay? Not today. What is this? So for different stages during apoptosis, you should use different methods to check apoptosis. Okay? At the beginning, the DNA won't fragment at the beginning because the DNA is inside the nucleus. It's protected by the nuclear membrane. You will expect DNA fragmentation happen 80 minutes after apoptosis is induced. Okay? We'll talk about the extrinsic method to induce the apoptosis. That will be time 0. Once you induce apoptosis, you have to wait for 80 minutes in order to see DNA fragmentation. Activation of caspase is earlier. You can evaluate the activity of caspase somewhere around after 30 minutes. For your NSM5 and PI flow cytometry, you need to wait until phosphatidylserine exposure. So that's something an hour later. So timing is very important. It will tell you when to use what method for your evaluation. Okay? We'll talk about some of those later. One of them is DNA fragmentation, which happens at the end of apoptosis. So we can assay apoptotic cell by looking at the phenotype of the DNA, looking at DNA ladder or the tunnel assay, tunnel assay. So let's look at the DNA fragmentation, because the apoptosis will activate protease. The protease will randomly digest DNA fragment, random, okay? If the DNA is protected by the histone, that will be protected. The endonuclease will not cut it at all. So normally, your DNA wrap around the histone complex about 180 base pair. And the linker is about 20 base pair from in average. Okay? So if the endonuclease get activated and randomly just cut all these DNA, you'll find all of them get digested into 200 base pair. And when you run it on an aqueous gel, it looks like a DNA ladder. That's the DNA ladder only appear at the beginning of the DNA fragmentation. At the end of the DNA fragmentation, everything gets collapsed into 200 base pair. Okay? So by looking at the proportion of the larger fragment, you will know at what the time point of the apoptosis. So that's time 0, 1 hour, 2 hours, 3 hours, 4 hours. See it? That's DNA ladder. And that's also DNA ladder. And you can use genomic DNA to do the test. Okay? So when can you do this experiment? If you are isolating some compound that direct damage the DNA or can cause apoptosis of the cancer cell, after treat the compound to the cell for like 2 hours, then you can isolate genomic DNA and run the gel. That's it, apoptosis. very easy to identify. Okay? Oh, I forgot. For the on the right hand corner, that's the DNA fragment on the DNA ladder, meaning your DNA structure have a nick, a broken section, okay?
Right.
So the nick is happening at the 3' end of your phosphate backbone, phosphodiester bond, right? So you should have a phosphodiester bond. And the new, if this is say DNA polymerization or replication reaction, the new DNA should be added on the 3' end, right? And added right here, 3' end of your sugar base, right? And so what happened in the tunnel assay is that we don't use polymerase for adding the fluorescent dye. We use a nucleic acid transferase, which only add one nucleotide once. It doesn't polymerize. Okay? Add one nucleotide at 3' end. So I can add the nucleotide at the broken 3' end. It should show fluorescent dye, right? So using this concept, that's a terminal assay. Terminal deoxynucleotide transferase, TDT, Dalton DUTP, that's the fluorescent that get hooked up at the end of the nucleotide. And it's going to label at the nick end. Nick means only one cut. One cut, okay? So expose the OH group, and you can have the transferase adding the new nucleotide. The new nucleotide should have biotinated DUTP that has fluorescent on the back. So you will know that DNA is broken. Okay? Why do you want to do that? If you have a fluorescent or a DUTP labeled fragment, if that's a brown color, you can do the in situ hybridization to see what cell in this tissue is undergo apoptosis. Okay, so this is about 30% of the cell in this tissue slide undergo apoptosis. So you can quantitate it. You can quantitate. This is not protein assay. It's labeling using tunnel assay. It looked like protein assay. Oh, so that's
It's not right.
huh?
It's not right. It's not protein assay. Right?
It's not. It's tunnel assay. It's labeling the nucleic acid. It's labeling the nucleic acid. See, I just used a different substrate to show it.
So
If you don't have a lot of money, you use the coloration to to show whether it's the labeling. If you have a lot of money, you use fluorescent. Okay, that's the differences.
So it only highlights the end, the first end and the end of the.
No. It highlights the 3' end that was nicked by the enzyme, by the endonuclease.
There's a
The
3'.
yes. The endonuclease, well, it can only add it on the 3' end. That's how replication is from 5' to 3' end.
Yes.
So your new molecule, 5' end, is going to hook up on the 3' end. So you have to expose the 3' end. Okay? But it's the phosphodiester bond that get nicked. One of the ester bond has two binding, right? And one of the arm is nicked.
Yes.
So that's expose the 3' end from inside of the DNA structure. It's just simple DNA replication reaction. Okay. So let's go. What time is it now? What? Let me finish. Let me quickly take a look. So apoptosis is a normal mechanism. It's not something abnormal or it's not something horrible. Okay, it's a normal mechanism for cell or an organism. So programmed cell death is important for development. We kind of know it already from Sidney Brenner's experiment. And then programmed cell death is needed to destroy cell life threat to an organism. Okay, so that's the two reasons for the development. That's Sidney Brenner's experiment using developmental C. elegans embryo. Okay, the 3 cell that is circle and pointed using the arrowhead, those are dying cells. In C. elegans, these are the normal cells, and those are the vesicle inside the normal cell. When the cell dies, it gets popping out, and it condenses into a smaller cell like this.
So it's visible.
It is visible. It is visible. Normally, each cell is looking like this, the same size, and when it's undergo apoptosis, the cell condense. Yeah, because you need to shrink it down in order to allow the macrophage to eat it. So that's a sign of apoptosis. So don't talk about this. For Dr. Horvath's experiment, the morphology of dying cell looks different. So anything that looks like popping out circle has mutation initial CED. Anything that is defected in engulfment of degradation, it looks different under the microscope, and that's how they get different name for that protein. Apoptosis is also important for limb development. So it's important for your digit formation. When you were born, you should have 5 fingers on each hand. But if your apoptosis, if the mechanism is defective, the skin between the digit is it's not removed or digested by your macrophage, then you will have whip-like, just like the
like a duck,
yes. But that's easy fix. Usually the surgeon will take a scissors, just cut it.
Look at the skin.
Yeah, just cut it.
What is grow?
Ooh.
Some of the
Grow.
some of the inherited disease, you will have the bone that is attached to each other. That's different from apoptosis. Yeah. So digit formation is important. The second one is ossification. In order to have a strong bone, you need to ask your chondrocyte to go dying, okay? Otherwise, and then we talk about the death of neuronal cell. So this is your heart bone. That's your young bone. Has a lot of lacuna of chondrocyte. This is the lacuna of your chondrocyte. And in order to have a strong bone like this, you need space for osteocyte to get in there and have the mineral deposit in there. So the chondrocyte will go apoptosis to create space for your osteocyte. Okay, so that's a must for ossification. And other example for programmed cell death and development is the tapeworm resorption. Frog.
Right.
Oh.
Tadpole becoming frog. And the nerve cell connection and formation of reproductive organ. That's interesting. If you don't have apoptosis of the female organ, then the male individual will have both reproductive sexual phenotype. Yeah. So for all the embryo, the reproductive organ started with female, and for the Y chromosome will give you the steroid that will cause the apoptosis of female tissue, and then stimulate the growth of the male tissue. Okay. So if you don't have apoptosis, or if your male hormone is not enough, yeah, the regression of the female tissue is not effective. You will have both female and male syndrome. Not syndrome. Phenotype. So, yeah, gay or lesbian is not psychological happen, okay?
So it's a lady boy, right?
Huh?
Bro.
Lady born?
Boy.
Lady boy.
Isn't lady boy, right?
It's
Lady boy is different. Lady
It's
boy
not
is
affected by
different.
the.
Lady boy could be an extra. You have an extra Y chromosome.
Yeah, actually.
That's different.
It's phenotype also. They have both.
Yes. Yeah. But some of the gay or lesbian is psychological.
Yeah, it's just
Yeah.
psychological.
Some of it is.
Yeah, some of them.
It's like only for the trigger, like a trigger, right? Like feminine side.
Yeah, it's just because of the
If-
hormonal.
If
and also
the male individual
Mhm.
has really pale skin and really smooth skin, that's not psychological. That's hormone, okay?
Yeah.
That's hormone.
What?
But what if the person, I'm like the individual, they have two phenotypes? What
Only
happens?
estrogen will give you very, very smooth skin and soft skin. Only estrogen.
Only estrogen.
Yes.
But if the person have those two phenotypes, what's wrong with that? I mean, like,
Nothing wrong.
oh, nothing wrong.
Well, the person was born this way.
I mean, like, what happened to those apoptosis? Is it like the failure or something?
The apoptosis may occur, but not at the right time.
Ah.
So that's why they have
Yes.
two phenotypes.
Yes. Well, because everything is regulated, so something must be wrong. Could be the mechanism of apoptosis. Could be something else. Okay. So in that situation, your case is
Your case.
is
It's not his.
the case you presented. it's the timing is wrong. Yeah.
That's
Okay. So, apoptosis in adult, one of the is the mammary gland. Stimulation of the pregnancy will cause proliferation of the mammary gland. After they have delivery of the baby, the hormone change. So you need apoptosis to allow all these mammary glands to get retract into the normal size. Yes. Also, for the prostaglandin, without the testosterone, it gets shrink. Apoptosis. Initiate the apoptosis process. So these are the terminology. Something too much will cause. If you have too much apoptosis, you cause tissue atrophy. For example, neurodegeneration or thin skin. If you have too little apoptosis, hyperplasia, that will cause cancer. Hyperplasia is causing cancer. So it's the balance of life and death. So your proliferation must be balanced with your apoptosis. The last one, after this, I'll call for the class. For the threatening, cell infected with virus, your cytotoxic T lymphocyte will kill the infected cell by inducing the apoptosis. So if the cell get infected by the virus, it's bad for the whole organism. The cytotoxic T lymphocyte will kill the cell and ask the cell to go apoptosis. Okay? Cell of the immune system, once this immune response is complete, the effector cell, effector T cell or effector B cell, okay, must be removed to prevent from attacking healthy tissue. So you can be inflammatory cell, inflamed all the time. It's bad for your system. Okay? So that's two situations under the threat. Okay, we'll talk about the rest of them. We'll talk about the mechanism, the pathway next week. Remember to come in here 12:30, okay? And I'll try to finish all the apoptosis in 1 hour. I'll let you go 1:30, okay? Okay, I'll see you next week.